Hello, in this video we will discuss about the adjuvant's mechanism of action to stimulate antigen presenting cell and how inflammation will occur. So in the previous video we was discussing about the adjuvants type and their mechanism, and in this video we will understand with the cellular pathway and during the injection. So in this way, if you want to watch the previous video, you can watch the link in the description below. So here is the vaccine, which that is basically contain antigen plus adjuvant. So this antigen is basically when the future disease will cause, so it means the disease will prevent through this antigen, and this vaccine will inject intracellular injection, and the number one is the depot effect antigen presenting cell recruitment, because alum is contained, so it means release slowly. So in this way precipitation will be occur due to the alum, and that is why this precipitation coagulation or something clotting like structure you can say. And after this basically inflammatory cytokines will produce. This inflammatory cytokine will recruit the antigen presenting cell or dendritic cell from the blood cell, from the blood circulatory system. So here I am drawing the blood circulatory system contain antigen presenting cell, which that is recruit through inflammatory cytokine like interleukin, and in this way the proliferation and move toward the infection site or inflammation site, because inflammatory cytokine are produced from the intracellular injection where the depot effect. So the antigen presenting cell recruitment through this vaccine containing antigen plus alum or antigen plus adjuvant. So on the second step is the PRR activation, is known as the toll-like receptor and node-like receptor agonist, which that will bind with the adjuvant site. After this binding recognition, it will stimulate the intracellularly the phosphorylation, and here is the nucleus which that's contain DNA of the antigen presenting cell. So the DNA is the transcriptional factor will bind after the phosphorylation will produce the interleukin 12 and 4. So this interleukin 12 and 4 will produce, and after this the endosome basically also form, and here is the node like receptor are present in the cytoplasm, and the rod like receptor are also present in the cytoplasm, which that is also triggered by the binding of the adjuvant site for the transcription factor to stimulate that antigen against this antigen. So after this, here is the toll-like receptor collectively to form endosome, and after this endosome formation, the processing of the antigen will be occur. This antigen separately processed through a endoplasmic reticulum, and in this way the antigen processing will lead to show the antigen through a MSC class molecule, MSC class 1 molecule. And let's here is that number 3 is the inflammasome activation through alum, and in this way the phagosome formation will be occur after the endocytosis, and in this way this phagosome will antigen processing will be occur. After the antigen processing, it will show through a MHC class 1 molecule in the surface of the membrane. So here is you can see the inflammasome basically will combine with a node like receptor P3 and ASC and caspase 1 protein. This will lead to inflammation due to the inflammasome formation, and here is the antigen processing mechanism will produce the MSC1 class molecule, which that show the antigen through this molecule, while here the inflammasome will produce the interleukin 1 beta, interleukin 1b, and inflammation cause. On the other hand, here the mediators can be possible to basically lead to stimulate the T helper 2 cell. But in this case here, let's begin to understand the naive cell will recognize this antigen through CD4 positive molecule through antigen binding with the T helper molecule, also the co-stimulatory molecule. I am not drawing, but just listen about that. And in this way, the naive cell, and after this naive cells, the step 4 is the immune cell presentation, MHC presentation, which that will lead to trigger the CD4 positive T cell, T helper 2 cell. This T helper 2 cell, which that will produce the interleukin 4, 5 and 13. This interleukin 4, 5 and 13 are also produced from the inflammasome. So in this way, the recruitment of the T helper 2 cell. While this T helper 2 cell and T helper 1 cell also the CD4 containing molecule, which that will show the antigen to the B cell for the humoral response, and in this way the neutralizing the antibodies. And in this way the plasma cell and B memory cell will produce. Plasma cell produce lot of antibody which that collapse the antigen. And here the interferon alpha, which that will also trigger the opsonization antibody through cellular response. And in this cellular response, the B cell will also recognize through T helper 1 cell. And after the T helper 1 cell, the B cell opsonization antibody, opsonization, as well as the after the opsonization, the cellular response will be occur due to the production of inflammatory cytokines from the antigen and the infected site or something, which that is the endocytosis through macrophage, and the CTL cytotoxic T lymphocyte, as well as natural killer cell. So that's all thing about the clearance of the antigen to produce antibody. So the B memory cell will produce, and also the plasma cell produce from the B cell, and this plasma cell produce a lot of antibody, which that and B memory cell is used for the future infection to quick response to clear the infection, while here is the antibody will clear through plasma cell. So what the meaning of adjuvants? So adjuvant release slow antigen from vaccine after the injection. Its mean prolonged stimulation lead to increase the adaptive immune system for combating future infection. So that is the important and major mechanism for the adjuvants, and I hope you make sense about that. If you don't know about the adjuvant, so please watch another video which that is I had made before this video. So thanks for watching. If you like this video, please make sure to subscribe, like and share this video, and I am making more handwritten tutorial about the biology and more knowledge wise. Thank